All right, today we are going to be working on this diagram in your notes packet. So if you haven't already, get out your notes packet. This is on the very back page. You'll see this diagram. At the top, it says transcription, translation, coloring page. So that's what we're going to be working off of. Feel free to pause this video at any time if you need more time to get something done or written or any of the above. You'll notice there's some instructions down at the bottom. We're going to go through those instructions together to make sure we're all on the same page. So the first thing it says down below is fill in the missing bases on the DNA, mRNA, and tRNA molecules. So bases, we mean the letters. So the first thing we have to do is figure out where is the DNA. When we take a look here, we know that DNA is double-stranded. It's a double helix. So that's going to help us find it. So our DNA is over here. This is our DNA. You'll notice it's got two strands. It's got a copy strand and a gene strand. The gene strand is going to have the letters that are going to match with the messenger RNA. They're going to have the code to make the protein. Copy strand is just the opposite letters. So what we need to do is fill in the letters on this gene strand. You'll notice it's a little weird. Normally we draw the letters on the inside. We're just going to draw them on the outside here. And the reason is because it'll make our lives a little easier when we're matching with the messenger RNA over here. So we have an A here, and we know DNA has what letters? DNA has A's, T's, C's, and G's. So A always goes with T. So I would write a T right here. Here's an A, I'd write another T, C, and G. So you're going to fill those in on your sheet. If you need a second, just pause the video here. The next thing it says is that we're going to fill in the bases on the mRNA. So remember, in order to get the message out of the nucleus, this gray part's our nucleus here, we have to turn the message into messenger RNA so that that message can leave the nucleus. Now, mRNA has different letters than DNA. Remember that mRNA cannot have T's. So anytime we want to write a T, we have to write a U instead. So let's take a look at what this might look like. So we've got a T and a T. We know that T always goes with A. So we're going to write an A and an A here. Sometimes kids get confused and they think they have to write a U here. Well, T goes with A, and I can write A. mRNA and RNA has A's in it. So write an A here. G goes with C, C with G. Now, when we get down here, A goes with T, but I can't write a T here, so I'm going to write a U. So here are the letters for the mRNA. And again, feel free to pause this video right now, and then you can write them down. Now, our messenger RNA has to leave the nucleus. So this gray part's our nucleus, and it's going to go to the ribosome so that we can make our protein. We're going to rewrite, this is the same messenger RNA molecule. It's just showing it moving from here to here. So we basically have to copy this mRNA molecule the same exact way over here because it's just the same thing. So we're going to start with this ACU, and we're going to write ACU here. So basically we're just writing all the letters from bottom to top over here from left to right, because as it moves out, it's just going to have the same letters in the same order. Now, once it's at the ribosome, the tRNAs have to come and bring the amino acids. So we've got our little tRNAs. They look like little T's. And the way that they know which amino acid to bring is by matching with the correct sequence of letters here. So tRNA, it's still RNA, so it's going to have U's, no T's. So here, when we have an A and we want to write T, we have to say, oh no, it's RNA. We can't write a T. We're going to write a U. So this first one, A with U, C with G, U with A. And so we're going to write those matching letters on all the tRNA. Now the next step, label where transcription and translation occur on the diagram. If you remember, transcription has a C and translation has an L. C comes before L, so transcription is the first one. It's what happens in the nucleus. So transcription is changing the DNA into mRNA. So we're going to just write that in the nucleus in that area over there. Translation is what happens in the ribosome when we're making proteins. So we're going to go ahead and write translation here. Next step, we need to label a codon, anticodon, and peptide bond on the diagram. Remember that a codon is a series of 3 letters that codes for the amino acid. And those 3 letters are on the message of messenger RNA. So remember, our code's originally in DNA, and then our code gets transferred to the mRNA. So 3 letters on mRNA. Now, I'm going to circle these 3. You could circle these 3, these 3, these 3. Technically, you could even circle them over here because this is our messenger RNA too. But I just chose these 3 because it was easier to do at the end, less crowded. Our anticodon, anti means opposite. So opposite codon is the letters, or are the letters on tRNA that are matching with it. So here's an anticodon. Now, did you have to label this one? No, you could label this one, you could label this one, or this one. So it's going to come and drop off the amino acids, and then this tRNA is going to go fly away. It's going to go away, but it's going to leave the amino acid, and it's going to have been connected to the other ones to make a protein. Remember, amino acids are made up of proteins. And the order of those amino acids is what determines what protein is being made. Now, the last thing that it says here is color in the boxes next to the words to create a key. Color each item in the diagram. I want you to pick whatever color you want for each of these and go ahead and color these. I'm going to show you with labels, but you are going to color them, okay? So our DNA, you should know our DNA. Our DNA is over here. It's double-stranded. Again, you're going to color this. If you want to add a label too, but pick a color, you're going to color both strands, and then you're going to color that box on the bottom. We have our mRNA. So these are our mRNAs, okay? We've got our ribosome. Our ribosome, I'm going to go to nucleus next. Nucleus is already kind of colored, so if you can just pick a color or you can just color it gray, the box gray that you already have. mRNA, we've got to color both of these because it's just showing it before and after. Ribosome is this big blob here, this whole big blob. tRNA, I want you to color all of them. So these little T's, don't color the puzzle pieces because remember the puzzle pieces on top are the amino acids. So you're going to work on coloring the rest of these. And then once you're done, you can put away your note packet. If you have extra time, feel free to do the optional practice on Google Classroom.